Yeah.
So, okay, hello everyone. Welcome to the international course for the reproductive immunology. Firstly, please allow me to briefly introduce the lecturers and the courses. So this course is jointly organized by Wayne State University School of Medicine, Rosalind Franklin University of Medicine and Science, and Huazhong University of Science and Technology. So to the best of my knowledge, this is the first international course focused on the topic of reproductive immunology, from the basic to the clinical aspects in China. Most importantly, we are honorably invited two famous reproductive immunologists to give us the wonderful courses. They are Professor Gilmore and Professor John Clark Kim. Professor Gilmore is the Scientific Director of CS Motto Center for Human Growth and Development, a Vice Chair of Research, John M. Mellon, John MD Enrolled Professor at the Department of Obstetrics and Gynecology in Wayne State University School of Medicine. And he is also the president of American Society for Reproductive Immunology and the pre-editor-in-chief of American Journal of Reproductive Immunology and the editor-in-chief of Journal Placenta. Another lecturer is Professor John Clark Kim. She is the NGSD Letterman Professor and also the Director of Reproductive Medicine and Immunology, Obstetrics and Gynecology Clinical Science Department, Chicago Medical School, at the Rosalind Franklin University of Medicine and Science. She is the President-elect of the International Society for Immunology of Reproduction, and also the editor-in-chief of Journal of Reproductive Immunology and associate editor of American Journal of Reproductive Immunology. This is the schedule for the international course program. This program will involve totally 8 sessions from the basic and to the clinical aspects of reproductive immunology. So Professor Gilmore will give us the basic reproductive immunology courses, and Professor John Clark Kim will give us the clinical course for the reproductive immunology. So after finishing the whole courses, the attendances will be awarded the certificate signed by the Professor Gilmore and the Professor John Clark Kim. So before ending my brief introduction about the course, I would like to acknowledge the International Exchange Center of HUST for financial supporting this program. So tonight, maybe the director of the medical office of the International Exchange Center, Ms. Haikun Wang, also attend this opening class. So welcome, Haikun. Okay, this is the end of the introduction. So I hope all of the attendants will enjoy the wonderful course. Okay, thank you. And let's warmly welcome Professor Gilmore. Thank you. So I will quit. Okay, you can start.
Okay, let me share screen. Yeah. You can see the screen, yeah?
Yes.
The presentation. Okay, so good morning and good evening, everybody. First of all, I want to congratulate Dr. Professor Liao and to thank also Professor Joaquim for organizing this incredible meeting. This is an experiment, really. I am not used to speak to an empty room. I still not get used to speak to an empty room. I wish I could see all your faces so to have a more interactive way. But as I said, we will do what we can, what we have. Hopefully in the near future, we will be able to, again, have physical lectures and physical course. But this is what it is. So what is my plan? I know that we have a lot of participants, and I'm delighted that we have this great number of people coming. I understand from Professor Liao that we have 50 people, and I'm sure we have multiple levels of knowledge in the area of reproductive immunology. Therefore, my plan, at least in this first lecture, is to do a very basic introduction. So again, I want already to apologize to those who already have a good knowledge of reproductive immunology for what I'm going to talk now, since I try to give us more basic concepts of reproductive immunology. Let me just go now to the PowerPoint. Okay. I mean, I have the screen, but I cannot see the screen. Good, let me go here. So the picture that you see here, it's a newborn with a complication that is called hemolytic disease. And this is one of the classical examples of an immunological response of the mother attacking the fetus. Here, but also this hemolytic disease, who has been proven that there is a cross interaction between the maternal immune system and the fetus, is also one of the few successful areas in reproductive immunology, because it is an immunological problem during pregnancy that it has been diagnosed, but also is an immunological problem that has been successfully treated. So this is one example of immunological problem. Another example of immunological problem during pregnancy is what we call the recurrent pregnancy losses or the implantation failure, where the mother has all the normal components, however, the fetus is not able to implant and the mother is not able to carry on the pregnancy. So when we look at these cases, when the mother is attacking the immune system, the maternal immune system of the mother is attacking to the fetus, or in cases, as I say, when there is a rejection of the fetus, we ask the question, why did your mother reject you? Why the immune system of the mother is not accepting the implantation and the fetus? And the answer is, a second, I think we have a, and the answer is because pregnancy is a very complex immunological condition. And that is what we're going to discuss. What we call a very complex immunological condition, because at the same time that the mother needs to accept this foreign body, we're going to talk a little about that, this foreign body that is the fetus, it needs also to be strong enough in order to protect the fetus as well as the mother against environmental factors such as infections. And we are living this pandemic of COVID-19, and we know exactly that women are pregnant and their immune system needs to protect in the case that there is an infection. And we will talk a little more as we reach the discussion about the infection and pregnancies. If there is anything, please feel free to interrupt me, or if there is a problem with the connection, also please just raise your hand or unmute yourself and let me know, okay?
Yeah.
So, there we go. I have to go back to the, okay, let's see. Yeah, perfect. So the history of reproductive immunology. The paradox, it really comes from almost more than 50, it's almost 60 years ago that a scientist in England, Sir Peter Medawar, who originally was born in Brazil, but he was doing the research in England, he recognized the paradox that the fetus, in genetic terms, is the semi-allograft that in principle should be rejected. See, Medawar was a transplant immunologist. So he was trying to prove that in nature, it's possible to take organs from one individual and put it in the other one. And he used pregnancy as the best example. He says nature is establishing in a transplant in a natural way. Why? Because each of us, each embryo, each fetus, each baby is made of 50% of the mother, but also 50% of the father. So in a way, those genetic components of the father represents like a transplant, a paternal transplant, the husband being transplanted into the wife, into the mother. So Sir Medawar saw that and came with the concept that the pregnancy is a semi-allograft. We will define what is allograft. And the maternal immune system will recognize those paternal antigens, and therefore the fetus in a natural way should be rejected if there was no mechanism that prevent that rejection. That is really the area of the reproductive immunology or immunology of the pregnancy, which focus on the concept that I mentioned just to you. Let me move this so I can see it. The idea of Medawar looking at the father, looking at the pregnancy, at the components of the father in terms of a semi-allograft. And since then, almost 60 years ago, the area of reproductive immunology has focused mainly in this aspect, that is the graft versus host response, where the mother is the host and the fetus is the graft, and how that interaction is maintained has been the main focus in the field of immunology of pregnancy. I don't know if you can see my pointer. Professor Liao, can you see the pointer, my arrow or no?
Yes, I can see. Yes.
Very good. Okay, excellent. Thank you. So, pregnant women are considered also and have been defined as a high-risk populations since, especially during pandemics. And COVID-19 was another pandemic, which immediately put attention on what is going to happen to women. Why we are caring about women during pandemics? Because one of the concepts that were introduced by Sir Medawar in terms of maintaining the pregnancy is that similar when we do a transplant, an organ transplant, we need to suppress the immune system. We will talk in a minute. So in order to have a successful fetal transplant, if we want to call it, the mother has to be immune suppressed. And the mother is immune suppressed, then she's in high risk of getting infections. And that is the reason for many years, and still even today, pregnant women are considered a high-risk population, since they are more likely to experience severe complications, mortality, and preterm labor associated with viral infections compared with the general population. Increased mortality during the pregnancy during infections has always been attributed to a maternal immune suppression. That has been a paradigm that even today is still, for many people, is the general concept. And I want you to remember, because we're going to discuss about this, the general old concept based on the observations from Sir Peter Medawar 60 years ago is that a pregnant woman has an immune system that is immune suppressed, that is suppressed. So a major question has been, and you will be discussed with Professor Joaquim, are many of these pregnancy complications like preterm birth, recurrent abortion, preeclampsia, and so on, the lack of immune suppression. So is a pregnancy complication of the related generation because there was no immune suppression? That's a question that we're going really to try to answer at the end of the course, when you hear both the basic science as well as the clinical evidence. And I would love to hear your opinion. So remember, this question, you will give us the answer as is really all the pregnancy complications due to the lack of immune suppression. So let's review a little the basic concepts of pregnancy, and then we're going to review the basic concepts of immunology. So in order that all of us are in the same page and we understand what we're talking about. So remember, at the time of implantation, you see here, this is the embryo, the blastocyst, sorry. This is the blastocyst who has implanted in the uterus. And the blastocyst has three components. This is the inner cell mass who will give origin to the baby, the fetus, and the baby later. And the external part of the blastocyst is the trophoectoderm. The trophoectoderm, when the blastocyst implant, as you see here, it will breach the surface epithelium of the uterus that is indicated here. It breaches this and penetrates within the endometrium. And in the stroma of the endometrium, which you see here, you have a lot of different types of cells, including a high number of immune cells. The stroma cells, as well as the immune cells present here at the implantation site, will start communicating with this newly implanted organ, the blastocyst, and they will create a network of cytokines, growth factors, necessary in order to heal the wound, so repairing the epithelium that was broken, accept this newly embryo, and also feed that the embryo will grow. So now, in terms of the reproductive immunology, here comes an interesting aspect, because the component of the blastocyst that is going to be in contact with all the maternal cells, including the maternal immune system, is the trophoblast, originated from the trophoectoderm. Is the trophoblast who is going to invade the uterus. Is the trophoblast who is going to be in contact with the maternal blood. And is the trophoblast who is going to be in contact with the maternal immune cells present in the decedent. Let me go back for a second. The interesting aspect is that the trophoblast, from a genetic point of view, or the trophoblast which gives origin to the placenta, the trophoblast that gives origin to the placenta, the organ who is going to function as the barrier between the maternal side and the fetal side. The placenta is made mainly of paternal organ, or paternal genes, Let me repeat that. The placenta or the trophoblast, who are the fetal cells who are going to be in contact with the immune system of the mother, are dominated by paternal genes. How do we know that? Because there are pregnancy complications such as the mola, and the mola is a pregnancy where there is an implantation, there is a placental formation, but there is not a fetus. And the mola is made mainly of two sperms who have fecundated an oocyte, and they took out the maternal pronucleus. So two paternal genes, two paternal pronucleus give origin to the placenta, but not to the fetus. And again, we know that by sequencing and so on, the dominant genes who are in the placenta are mainly coming from the father. So looking immunologically, I like this picture, is this part outside, this represents the placenta, who is made of paternal antigens. And the fetus, the baby, is made of both paternal and maternal. Now, if you look at this picture, I always ask, what mother will reject such a beautiful baby? There is no mother who will do that. However, things coming from the husband, things coming from the father, maybe. And the placenta is coming from the father, maybe that is the source of some of the reactions. So based on this first concept that I'm giving you, really we need to define the immunology of pregnancy, because the trophoblast are the cells that will be in the maternal circulation, and they really are going to be dealing with the immune system of the mother. The trophoblasts, as I mentioned, are the cells that will invade the uterus and will trigger a local immune response. Therefore, we really, when we talk about reproductive immunology, we are not really, or the immunology of pregnancy, we're not talking about maternal fetal immunity, because the fetus is protected. We're really talking about maternal trophoblast immunity, how the maternal immune system is interacting with the trophoblast, with the placenta. And as I mentioned to you before, we say the pregnancy is a unique immunological condition. And why is that unique? Because if we look now carefully, that has been done for many years, characterizing almost all the cells present in the implantation site, in the endometrium, or later known as the deciduum. A major dominant cell type present in the endometrium and the deciduum are immune cells. And you have all the immune cells that we know, all of them. 70% are NK cells, 20% are macrophages, 10% are T cells, including B cells. For many years, we thought that in the endometrium, in the implantation site, there was no B cells. Now we know that was incorrect. B cells are present there. There is a different type of B cells. We will talk about the immune cells, but all the immune cells are present on the implantation site, both native as well as innate immune cells.
So, Professor Gill, we cannot hear you. No sound.
What about now?
Yes.
Yeah.
Okay. How much did we miss?
About 30 seconds.
30 seconds? Okay, thank you. So let me go back again. I think I clicked by mistake. Thank you. Let me repeat this aspect. That I say that the pregnancy is a unique immunological condition, because as I mentioned to you, immune cells, all the immune cells that we know that exist are present at the implantation site. And the fact that we found those immune cells at the implantation site was a support for the idea that the maternal immune system is recognizing this foreign body that is the fetus, and consequently is attacking the fetus. Because we know that recruitment of immune cells only happens to sites where there is an infection or when there is the presence of a foreign body. So here you have the presence of a foreign body that is the fetus and a recruitment of all the different maternal immune cells. Again, suggesting that the mother is recognizing paternal antigens, or the maternal immune system is recognizing paternal antigens. They are migrated into the uterus and they are trying to attack this semi-allograft that is the fetus. Good. So this is the definition of what is happening during the pregnancy. Let me give you a quick immunological review for those who are not immunologists. So again, just to give you we can talk the same language, if I can say that. The classical definition of the immune system is the body response to foreign material. That's the definition. The immune system is patrolling our body, trying to identify in the moment that something foreign will invade our body. And the way it does is by having two major systems. One is the innate immune response, and second is the adaptive immune response. Both are evolutionary different. The most older in terms of evolution is the innate immune response. And the cells who are protecting are mainly divided in what is called the granulocytes and the monocyte macrophages lineage. And in the later, or the adaptive immune system, which evolutionary is relatively new, is mainly done by B cells and T cells. So if we look at the innate immune response, we have this family of macrophages. They are originated from the monocytes, and the role again is to clean up the tissues as well as clean up of bacteria or viruses that may be penetrating the tissues. Granulocytes, who are in the circulation and the tissues, they are the killers. They have the bombs that will bombard any infected cells. You have the natural killer cells, the dominant cells at the implantation site, and you will hear from Professor Joaquim a lot about NK cells. You have the dendritic cells, the classical antigen-presenting cells. Again, all of them, what they do is they patrol, they phagocytize, and they kill any foreign body that is going inside of the tissues. The adaptive immune response, as I mentioned to you, is by T cells and B cells, but based on the type of signals coming also from the adaptive, so the innate immune response, you can divide it in two types. One is a cellular, made of the cytolytic response, dominated by CD8 T cells, CD8 positive T cells, and a humoral response that is antibody producing cells. That is the classical, dominated by B cells. This too has been called the Th1, Th helper 1 against cytokines, pro-inflammatory cytokines, and Th2 anti-inflammatory cytokines, which is defined here. So the Th1 is dominated by IL-6, interferon gamma, TNF-alpha, increased cell-mediated immune responses. Th2, an example is IL-10, interleukin-4, increased humoral response, production of antibodies. This is important because there is another concept that has been around for many, many years, and that is that pregnancy represents a Th2 environment. In other words, the non-pregnant woman is in an inflammatory environment, But the moment that she gets pregnant, she becomes in an anti-inflammatory environment. And again, that was a concept, and we're going to discuss a little more later, who has dominated also the field in terms of this dichotomy, pregnancy Th2, non-pregnant Th1. So let's go back now to the fetus and the allograft. This is a transplantation course 101 in 10 seconds. So if you take a piece of skin from yourself and you put it in another part of yourself, that is called an autograft. And we happen a lot today, especially for if there is burnings or even cosmetic surgery. If it's in the same individual, the autograft, the outcome is no rejection. If you take a piece of skin from one individual and you transfer to another individual, this is what is called an allograft. And in these cases, the outcome is always a rejection. And there is another area that is called, if you take an organ from another species and you put it in another species, that is called xenograft. We know today, for example, takes the bulbs from the pig and put it in humans. That is coming from one species to another species. It's a xenograft transplant. That is also is associated with rejection. Both allograft and xenograft, in order to succeed, you need to induce what is called an immune suppression of the host. Allograft, of course, you don't need immune suppression. There is no rejection. Pregnancy has been considered in this area, an allograft. And this is the classical experiment that Silmeda and others almost 60 years ago did. If they will take a piece of skin from a black mice and will put it in a white mice, they will be rejected. However, if they made the two strains of mice, and when the mouse was pregnant, will do the transplant of the skin, that a rejection will not happen. Now, if you will repeat the experiment using different mice, I repeat, again transplanting the black into the white, you will have again the rejection. So based on this observation, Sir Medawar and many others concluded that something is happening during the pregnancy that is preventing the rejection of the paternal antigens. And the question has been, what are those factors, or what is preventing that rejection? And of course, when you have such an interest, clinical and scientific observation, you come with a lot of hypotheses. And I want to do is to review with you just 4 hypotheses, who again still are, you will still hear them, and we will see what is the pros and cons on those hypotheses. One is the mechanical barrier. Second, suppression of the maternal immune system. Absence of MH class 1 molecules. I don't think we will discuss much of this. Type 2 immune responses and local immune suppression. Let's start with the more classical, the mechanical barrier. It has been a concept in the area of what is called immune privileged organs, that they are separated from the rest of the body. What are the immune privileged organs? The immune privileged organs are those related to reproduction. What is the first and more important reproductive organ? The brain. And the brain is separated from the rest of the body by what was called the blood-brain barrier. And we always thought that nothing goes into the brain in terms of immune cells, bacteria, and so on, cytokines, and so on, and nothing comes out from the brain. So a nice barrier separated into components. The ovaries also separated, the testicles in the male also separated, and pregnancy was thought to be separated from the mother by this mechanical barrier that is the placenta and the fetal membranes. However, we know today that immune privileged organs have nothing to do with the mechanical barriers. And in reality, that idea that nothing is moving between privileged organs and the rest of the circulation now has been well proven that is wrong. Those barriers exist and they are there, but they are not impermeable membranes or impermeable barriers. They are permeable barriers. They are controlling the flow. They are not preventing the flow. And that is very important differences between controlling the flow versus preventing the flow. There is no prevention. There is a control of movement. So when we go to the pregnancy, the placenta and the membranes, we know now that the concept that nothing comes out from the fetus or something goes to the fetus is not true. There is evidence for traffic in both directions across the maternal fetal interface. And what do I mean with that? We know that cells from the fetus are continuously migrating into the mother. And that is not happening only at the time of delivery. From early stages of the pregnancy, or even at the time of implantation, fetal cells start migrating into the maternal circulation. And they have an early contact with the maternal immune cells, number one. Number two, those fetal cells, they are first in the circulation, but also then they go and will penetrate the maternal organs. And they integrate into the maternal organs. And they will differentiate into the cells of those maternal organs. We know, for example, that fetal cells can go to the liver. And when seed within the liver, they will differentiate into hepatocytes. They can go into the mammary gland and they will differentiate into mammalian cells, and so on and so on. And the interesting thing is when those cells go into those organs, they provide a better capacity of repair. They help the mother for repairing their tissues. So I always like to say that is a nice way how the fetus is contributing, is giving back to the mother by helping her tissues to repair. There is another important aspect in terms of the immunology, because that it means that the fetal cells are not within the mother just for the time of the pregnancy. In other words, the immune tolerance to the fetal cells doesn't happen.
胎儿胚胎作为
I missed the message. So if it's nothing important, I will continue. If not, Professor Liao, correct me, what is the message? So what I'm saying is that the fetal cells who are now present in the tissues of the mother are going to be there present for the rest of the life of the mother. So the mechanism by controlling the tolerance are not only from the time of the pregnancy, are necessary for the rest of the life of the woman. And that is critical. And I always like to put here an important message for the young women who have not married and are looking for a husband. Make sure that when you look for a husband, you choose carefully, because you will be carrying the genes of your husband for the rest of your life. So make sure you choose carefully your husband. Let's move now. So clearly there is no immune barrier. So we cannot explain the lack of rejection during pregnancy based on a mechanical barrier. I forgot to tell you also, there is maternal cells going into the fetus, T regulatory cells, macrophages from the mother goes to the fetus, integrate into the immune system of the fetus, and they are the first educators of the fetal immune system. The maternal cells are helping the fetus to acquire the process of self-tolerance and so on. So education from the maternal side to the fetus starts very early during the pregnancy. So suppression. The concept that I mentioned to you early, that the maternal immune system is immune suppressed, and that pregnancy is characterized by a state of immune suppression, systemic immune suppression. So similar as a graft, the maternal immune system is suppressed during pregnancy, allowing the semi-allograft to implant and grow. That was the concept that has been around for almost 60 years. But then it comes the question of reality. A mother that is immune suppressed will be understandable in our society when the mother lives in a clean house, is protected, delivers the baby also in a sterile environment. But if we look through history, through evolution of our species, or if we look also in 80% of the world, they do not live in sterile environments. They live in conditions who are immunologically challenging. The mother, when is pregnant, she is working in the fields. If she needs physical needs at the bathroom, she will go to the side, do her needs, clean herself, and she will not wash her hands. She will live with the same hands. If a pregnant woman will be immune suppressed, she will not survive. Moreover, we as a species, we will not be here today if the pregnant woman was immunologically suppressed. And the concept is not new. This is a painting from the 1700s, and I love this painting. Here you can see a pregnant woman and a man eating, and here is another woman taking the food and sharing between all of them. Just imagine this pregnant woman will be immune suppressed. She will be dead, and we will not be here. So the idea of a maternal, and this is a more recent picture, see a pregnant woman, very pregnant, not only that is working on the fields, she's strong, she's carrying things, she moves around, she's not isolated in the house, she is in the environment. And I will repeat, she's very strong, she's able to carry everything. It's not just a physical strength that this pregnant woman has, also she has an immunological strength, because she needs to protect the species. She's the one who is keeping the species and the future members of the species. So she needs to, the immune system needs to protect the mother and needs to protect the fetus. Consequently, we conclude in this, the message to you is, the maternal immune system during pregnancy is stronger than ever, because it's an evolutionary demand that it has. The suppression of the maternal immune system also doesn't make sense at the times what we convey with pandemics. We know that maternal antiviral immunity is not affected during pregnancy. HIV pregnant women do not suffer from AIDS. Moreover, during the pregnancy, they are protected against HIV and they don't die of AIDS. And now, all of you have heard and are following that the mortality of pregnant women with SARS-CoV-2 is very low compared to the general population. So if a pregnant woman will be immune suppressed, they will be dying with in the time of pandemics. There is another reasons why they died, and we will talk in a future lecture when we discuss about infections and pregnancy, why there are some cases where there is a higher degree of maternal mortality with viral infections. That has nothing to do, and I repeat, it has nothing to do with an immune suppression. The next aspect is the pregnancy as in unique immunological conditions, as we saw, let me press this, the presence of immune cells. Why you will have so many immune cells at the implantation site? As I mentioned to you, these immune cells that are coming into the uterus, all these immune cells, was thought for many years that they are attacking the fetus. They were used as the evidence that the maternal immune system is recognizing the paternal antigens and wants to reject the fetus. The interesting thing, my lab, as well as many other ones, have worked on all these immune cells. And our approach during the years was, if we deplete of immune cells, we will help the pregnancy. Again, let me clarify. If the hypothesis is that the immune cells from the mother are attacking the fetus, the best way to protect the fetus for rejection or any immunological complication is just delete the immune cells. Remove the immune cells from the uterus. And that's what we did, we and many more other ones. And here is the surprise. Depletion of immune cells at the maternal fetal interface, instead of helping the mother, instead of helping the pregnancy, it terminated the pregnancy. So had detrimental effects to the survival of the fetus. So in other words, the immune cells at the implantation site are necessary for the success of the pregnancy. We need each of those immune cells for a successful pregnancy. And we will talk more in detail of each one. So wrapping up this part, we challenge this concept that the pregnancy is a graft versus tumor versus is the graft versus host. There is no doubt that there are mechanisms of induction immune tolerance, because I told you, the immune system of the mother needs to accept the paternal antigens for the rest of her life. So it needs to have mechanisms of paternal tolerance. But they are not associated with the classical graft-host immune responses. It's not associated with suppressions or immunological suppressions. We think it's more associated to the immunological changes that we see during tumor development. And again, it's very interesting how much we have learned from tumor immunology that may have a better application to the immunology of pregnancy. So let me show you some examples why we think is the case. When you do an organ transplant, such as a kidney that you see here, you are transplanting a full developed organ into another individual. When you transplant this organ, you're going to have a massive release of proteins or antigens from this fully developed organ. In addition, as in any organ, you have dendritic cells, immune cells, but especially dendritic cells that are present within that transplanted organ. And although there is a lot of discussion of these donor-derived dendritic cells, there is a lot of work showing that these donor dendritic cells can take many of the antigens of this transplanted organ, for example, the kidney, go to the lymph nodes, present to the T cells, trigger an expansion of cytolytic T cells, and then you will have a rejection, a classical rejection. When we look at the tumors never start as a big mass. They always start with a few cells. We call them the cancer stem cells, the tumor-initiating cells. The interesting thing is that these tumor cells, when they start growing, start releasing proteins from the malignant cells. And the dendritic cells start taking this small amount of proteins. They go into the lymph nodes, and they do what is called a small antigen presentation, a way to induce tolerance. We do that for allergies. If you give to a child with allergies, for example, peanuts, if the child has peanut allergy, you start giving a small amount of peanuts and you start inducing tolerance. And it's based on the concept of a small amount of antigens are incapable of triggering an immune response. They at the end will induce a T cell exhaustion or deletion of clones to represent those antigens. That's what we call the induction of tolerance. Pregnancy. I never have seen a pregnancy that starts with a full developed baby. Never. All the pregnancies that I'm aware always starts with 2 cells, and then 4 cells, 8 cells, and so on. So in the early stages of the pregnancy, the morula and the embryo started releasing fetal antigens containing paternal proteins, paternal genes. And exactly as in the tumor induction of tolerance, dendritic cells present at the implantation site, they will capture those small amount of paternal antigens. They will go to the lymph nodes. And again, the small amount of antigens that I mentioned to you in both cases will induce to the deletion of effector T cells, expansion of T regulatory cells, and the induction of a specific paternal antigen tolerance. So again, it's the induction of tolerance, very similar to what happened into the tumor, is very specific and is organized in a way that will have a long-term effect. In terms of the immune cells, we go again to the tumor. The tumor secretes factors that educate the immune cells that are present in the tumor microenvironment. And we know now, for example, that macrophages or tumor-infiltrated macrophages, they are helping the tumor to grow. And there are a lot of T regulatory cells inside of the tumor. And there is also NK cells without the tumor. And they are not able to reject the tumor. They are not able to kill the tumor. The tumor escapes this immune surveillance by secreting factors that controls the immune cells, that educate the immune cells within the tumor, such as PD-L1 and FAS ligand, and all these checkpoints who are critical for controlling the local immune cells. That is exactly what we see in the uterine microenvironment. The placenta, and we will discuss in one of the classes what is the specific factors. The placenta is secreting factors who are able to modify, all the immune cells who are present at the implantation site. What do I mean modify? They change their characteristics, their behavior. They educate them into protectors, into supporters. That's the right word. They become supporters of the pregnancy. And we know that macrophages are critical for trophoblast invasion, NK cells are necessary for spiral artery transformation, dendritic cells are necessary for implantation, and so on. So to summarize this, pregnancy is characterized by the existence of a specific cellular and molecular mechanism that tightly regulate the immune landscape without compromising tolerance. There is a tolerance, but the immune system needs to be active and strong. The fetus, the placenta, secretes a lot of factors that are controlling, educating those functions. Let me go now to the infection, and that will be the end of my presentation. What I mentioned to you, pregnancy has been defined as an anti-inflammatory condition, and the presence of inflammation has always been defined as a breach of tolerance. So inflammation was always been thought to be bad to the pregnancy. And that was what is what's called the Th1, Th2 hypothesis. However, there has been a lot of, again, pregnancy as a Th2, Wember suggesting the hypothesis. However, there has been a lot of contradictory studies, looking in early study and early pregnancy and so on. And we did a small study looking at cytokines in a non-pregnant and the pregnant woman. And we look at the first trimester of the pregnancy. And for our surprise was that in the first trimester of the pregnancy, compared to the non-pregnant, there was a lot of inflammatory cytokines. And then when we look in the different trimester, it was the interesting thing because the first trimester had a lot of inflammatory cytokines. However, when we went to the second trimester, those inflammatory cytokines decreased, exactly as was reported. Yeah. However, in the third trimester, they came back. So let me put you in a figure where now define how is the pregnancy in terms of inflammation. The first trimester of pregnancy is an inflammatory process. You need inflammation in the first trimester. And in the next class, we will talk about why inflammation is necessary. The second trimester is what we call the Th2. Yeah. And then we know if we see the woman in the first trimester, she has all the symptoms of an inflammatory process. The fever, even fever, tiredness, she feels terrible, everything is not, all those are not just hormones. We always blame on the changes of estrogen and progesterone. Clearly, it's not just the estrogen and progesterone, it's also the high levels of inflammatory cytokines that are affecting the early stages of the pregnancy. The second trimester, the woman feels fantastic. Why? Because indeed, this is a time of growth. This is a time where the inflammation has gone. This is the time of this anti-inflammatory environment, which is helping tissue renewal, feeding the growth, and so on. Is a perfect time. And the last part of the pregnancy is again inflammation. Inflammation that is required for the process of parturition. Without inflammation, there is no parturition. So again, the immune cells are also changing in each of these stages. So pregnancy is not just a single immunological condition from the beginning to the end. It's a continuous evolving and changing, which is what it leads to the successful pregnancy. To put it in other words, so we started, contrary to what we thought in the past, we start with really an anti-inflammatory condition of the non-pregnant woman. The implantation time or the early first trimester, now we know that it's only the early first trimester, is pro-inflammatory. Then you need a shift from this pro-inflammatory into this anti-inflammatory condition. This is a critical stage, evolutionary. We will discuss in the next lecture. I repeat, this is a critical evolutionary condition, going from the pro-inflammatory into the anti-inflammatory. And this is the stage that we say is associated with the success of, with the growth of the fetus. And then the last shift, that's for the parturition, which is an inflammatory process. The major questions that now we are working and trying to understand, what regulates these shifts from the anti-inflammatory into the pro-inflammatory, and from the pro-inflammatory back into the anti-inflammatory. Yeah. What is the signals that shift? Why this is important? Because the signals of the shift, or the shift on the signals, is what maintains the balance between the implanting fetus and the immune cells. If there are abnormal changes, the same cells who are helping the pregnancy, now they will be responsible for the rejection. So many of the hypotheses that are working, for example, is that the placenta, the fetal signals, will change an inflammatory macrophages into an anti-inflammatory macrophage, and then will become again a pro-inflammatory for the parturition. Let me see. So the normal pregnancy is characterized by those shifts. When the shifts occur at the wrong time, you have the pregnancy complications. So if the inflammation comes too early, that is called preterm birth. If the inflammation never stops and you continue the inflammation, that there is not a shift into anti-inflammatory condition, you have early pregnancy loss. However, if there is no inflammation at all, or if there is excessive inflammation, you will have what we call implantation failure. And with that, I will stop, and I will be happy to answer any question that you may have. If you have any question, please feel free to unmute yourself. Professor Liao, do we still have some time for questions?
Yes, we still have time.
I hope I was clear enough for everybody. I know that the language is a challenge. The distance, not seeing each other, is also a challenge. But I will be happy to answer any question.
Hey, Dr. Gilmore.
It's only for the students, the question, Dr. Salih.
I'm worse than the student, you know that, right?
The students from China. Anyway, Professor Liao, do you allow some external people to ask questions?
Yes.
Well, thank you so much. My question is, you drew the comparison between the cancer and tumorigenesis and the early pregnancy, and how they both seem to modulate the immune response in a similar fashion. Now, if we talk about pregnancy complication and pregnancy loss, do you think the same mechanism can be used to actually combat cancer and tumor growth?
Yeah, and that's very important. Exactly. Many of the things that we learn from the cancer now, we're really seeing that is happening in the pregnancy, as I mentioned. But now, many of the things that we are learning from the pregnancy complications, we try to use into the cancer. For example, the work of blocking changing the Th2 into Th1 into the tumor, who has been the basic concept of the tumor immunology. We knew how difficult it was in order to really induce anti-tumoral response, because you have so many M2 macrophages who were stopping everything, and the famous PD-1, PD-L1. Yeah? This is the classical example of an immune regulation by the tumor into the blocking the immunological, blocking the immunological surveillance, sorry. Again, the whole concept of blocking this PD-1, PD-L1 has led to incredible and successful immunological treatments into the tumor. Professor Liao has shown in several papers, the same with Dr. Joaquim, that many of these factors, PD-1, PD-1, and others in the family, are abnormally expressed during early pregnancy, which may explain also some of the complications like recurrent miscarriages, early recurrent miscarriages, and so on. So yes, the answer to your question is many of the abnormal regulations that we can see in the tumor immunology, many of the complications of the pregnancy, we can now transfer to try to reverse in the tumor. In the tumor, we want to end the tumor. In the pregnancy, we want to maintain. So we want to reestablish the immunological balance in the pregnancy and stop that immunological balance in the tumor. So yeah, there is a lot of, many of the components that we see in the tumor, again, PD-1, PD-L1 is a classical example. Fast ligand, another example. Galactin-9, all those immune checkpoints who are in the tumor are also in the pregnancy. Thank you, Dr. Sally. We give him a little time to the students to get courage in order to ask a question. There is no stupid question, so feel free to answer question. You can ask in Chinese also. Okay, so if it's okay, I will stop. Let me see. First, I need to stop the recording. No, sorry. Oh, Patrick, do you have any question?
Yes, I have a question. Thank you for your talking today. It's always a pleasure to follow you because there is some update. However, as sometimes there is some need to be clarified on some point. Today you introduced in your talk the concept of placenta has barrier which control the flow between mother and fetus. And you state that there is fetal cells which cross the placenta and go to maternal circulation. And you even mentioned that the tolerance from pregnancy is maintained even after pregnancy. So there is a few questions there.
Okay.
You clearly explain that, like in tumor microenvironment, during pregnancy, there is some substance which educates those immune cells and induce the tolerance. Since after pregnancy, we do not have any more placenta, how can you explain the maintenance of that tolerance? This is the first question. And
okay, so let me answer the question, because remember, I have Alzheimer's, so I don't remember. I will not remember your question.
Okay.
It's a joke. It's a joke. Thanks God. Top. I don't have Alzheimer's, but I forget questions. So let me answer to your question. It's a very good question, a very important question. So my friend, and to all of you, there is something that is called immunological memory, which is a growing field. When we talk about immunological memory, we don't refer only about the T cell memory or any associated with the classical memory T cells. We're talking a lot about what is called the tissue memory. So, and this is very important that you remember. Even the decidua or the uterus, it has what is called a memory. So the development of a good pregnancy is already a major step for the future pregnancies, because the endometrium will never be the same for subsequent pregnancies. They will have a memory how to function. The second pregnancy, in the majority of the cases, if there is nothing else, it will be easier. It will be, the adaptation will go faster. Even parturition, you know, the first parturition is the most painful. They can go for 14, 20, 48 hours, the parturition. Once the organs learn how to behave in those conditions, they will maintain a memory. And the next pregnancy, even if it happens 2, 3, 4 years ago, that parturition will be easier and faster. There is an incredible system that is called the tissue memory. The same applies for the immunological system of the woman. And I think that's the work we're doing with Dr. Professor Liao. The immune system of the mother, when the pregnancy ends, is not true that comes back to be as it was before. It will never be as it was before, because it needs to maintain a memory. And that memory, so the second pregnancy is already prepared. Now, why do we need a tissue memory or an immunological memory? It's evolution. When you do something good, you are already prepared. So the second one, you will save energy. Evolution is saving energy. So the memory gives you an evolutionary advantage. When there is no a tissue memory or a system memory, that species, from an evolutionary point of view, now is in a bad situation, will demand additional process of adaptation. That species may be disappearing. So tissue memory, organ memory, is an evolutionary adaptation that prepares that the second time everything is more functional, is better, and less energy. Yeah. So, long answer to your question. The changes in the immune system of the mother do not end with the end of the pregnancy. Those changes will remain forever, for good and for the bad. A bad pregnancy will create a bad memory. And that bad memory will increase the risk of having again pregnancy complications until the problem is fixed. So a preterm birth in the first pregnancy, high risk for the second pregnancy. If you get a second pregnancy that is good, the risk of the third pregnancy, a preterm birth decrease, and may have now a term. Again, is a very evolving. There is a lot of plasticity in this tissue system, that tissue dash system memory. Yeah?
Yes, okay. I get it perfectly. It answers in a part of my second question, because I stay at the same point. You just develop and talk about the maternal side, you know, with that immunological memory. But you have to clarify to me also about the paternal antigen. We know that each individual is genetically different from other. If a woman have several kids, do these paternal antigen will induce the same immune response? Because the memory, the tissue memory, was for, specific antigen. But now we have several pregnancy. What happened in this case? And that's
okay, so let me ask you a question. Are you referring if they change the husband or the same husband?
Even the same husband. I'm talking about the same husband.
Okay. Sorry, so what was your question?
The question is about the fetus, the future pregnancy.
Yeah.
Do the paternal antigen will be considered the same?
Yes, absolutely. The paternal antigen will be the same if it's the same father. If it's the same father. If it's the different father, you start going back to ground zero. Yeah. So it's interesting. I did a small study in Colombia where we were testing the idea of, is a pregnancy, a preeclampsia, is subsequent pregnancies high risk of preeclampsia? And it was very interesting indeed. If it's the same father, the next pregnancy, the risk of preeclampsia was significantly decreased. We never published that, we have to publish that. The numbers are not very big. However, if there was a change in the father, it's like going to zero. So if they have a new husband, the first pregnancy, they have preeclampsia. The second pregnancy with a new husband, they will not have preeclampsia. Yeah. Again, there is a flexibility based, always start changing. Always the adaptation process will have. If it's the same husband, so they did the effort in the first pregnancy, they established the memory, they established the tolerance. The next pregnancy, evolutionary, will be easier. There is no complications. If they change the husband, you go back to square zero. So you need to establish a new tolerance. If it has high risk of preeclampsia because of the environmental factors, they will get preeclampsia. With the same husband, the second pregnancy, or the second husband, the second pregnancy, no preeclampsia. Yeah? That's an example of what we now define as the immunological memory of pregnancy. Did I answer your question?
Yes.
Yeah.
But as you know, we state that.
Good. How do we know? We don't know. We don't know. We don't have this is the area that I think is very important now to understand at the molecular or cellular level, how is the establishment of the tissue memory and the immunological memory.
Okay, thank you.
We don't know.
Thank you, Professor.
The tissue memory exists. I mean, there is a lot of studies now showing tissue memory. There is tissue memory in the lungs, there is tissue memory in the heart. Many of those tissues have been shown to have what is defined the tissue memory. And more studies are coming showing a specific cell memory, even for immune cells. So it's a very interesting and fascinating area. It still is not clearly understood all the different mechanisms, how that is established. But we know in pregnancy that there is a long-term immunological memory.
Okay,
you can ask Professor Liao about that.
Yes, I have an idea. I have an idea. Yes, thank you. Thank you, Professor.
Okay. What time is now? Any other question? Professor Liao, should we stop now?
No, maybe I have no extra questions.
So, I mean, should we stop now or we wait for any other question from the students?
If no more questions from the students, then we can stop the this class.
Very good. Again, I want to thank you for today. It would be great if you give us a feedback of how was, at least it will help me, how was the imaging, especially with the cameras, if it was good enough, and also if you were able to understand. That will be very helpful. Okay?
Okay.
Beautiful. I'm looking forward for tomorrow lecture with Dr. Professor Joan Joaquin.
Okay.
Thank you.
Okay.